In this video we'll be looking at dihybrid crosses. So we have looked at monohybrid crosses in previous videos. So mono meaning one, so we looked at one trait that was passed on by the parents. In dihybrid crosses, di meaning two, we are going to look at two traits that are passed on by parents. Now, according to the exam guidelines, you need to know the following. You need to know Mendel's principle of independent assortment. You need to know the dihybrid genetics problems. You need to be able to do some genetics crosses. And then you also have to be able to calculate the proportion or ratio of the genotypes and phenotypes. Looking at Mendel's law of independent assortment, it is the alleles of two or more different genes that get sorted into gametes independently of one another. So, in other words, the allele a gamete receives for one gene does not influence the allele received for another gene. Now, if that doesn't make any sense to you, let's look at this diagram over here. So you'll get a chromosome from the paternal side as well as the maternal side, so one from the father and one from the mother. Now on these chromosomes, one chromosome can have quite a few different genes represented on it. So in this case, this gene over here would be, for example, for hair color. And that is the locus of where hair color is found. So on this chromosome, you'll find the gene for hair color, but then the locus for hair color will be exactly on the same spot on the mother's chromosome. On this chromosome, you can also have eye color that is represented and multiple other genes. So basically what Mendel's law is saying is that the gene or the trait for hair color will not influence the type of eye color that the person will have. So looking at the pictures on the right, if somebody has blue eyes, they can have blonde hair. But if somebody has blue eyes, they can also have dark hair. They don't necessarily have to have blonde hair. Blue eyes is not always connected with blonde hair. Same with curly hair. For somebody to have curly hair, it doesn't necessarily mean that they have to have dark hair. They can have blonde hair that is also curly. They can have blue eyes, dark hair, curly hair. Or they can have blue eyes, blonde hair, curly hair. Or red hair, curly hair, brown eyes, for example. So these traits do not influence each other. That is the basics of the law of independent assortment. When we look at dihybrid crosses, you need to know that there are 16 possible outcomes for the cross of these parents. So let's use, for example, Mendel's pea plants again. Yellow seed is dominant over green seed, and then a smooth seed is dominant over a wrinkled seed. So these are the two traits that will be crossed in this dihybrid cross. Just so if you're asking why a capital G is used and not a Y, some people's capital Y's look very similar to the lowercase y, and if they are on their own, there's no way that somebody will have a reference frame for how large that is. So it's very difficult distinguishing between the two. So having or using a letter where there's a distinct difference between the dominant and the recessive will be very helpful. Same with the wrinkled seeds, Y, and smooth seeds, Y and R is used and not an S. What do they want us to do? They want us to cross the homozygous dominant characteristics with the homozygous recessive characteristics. So they're telling me that all of the dominant characteristics have to be shown and they're going to be homozygous. So I've got a yellow seed with the trait of being smooth going to be crossed with a green seed that is wrinkled. So that is what that means over there. Now they want us to use the F1 generation to produce the F2. So to get to the F2, we first need to see what our F1 is, and that is where this cross comes in. So let's do this. P1 phenotype. Now when you write the phenotype as there are two traits, you can just very simply write it like yellow and smooth being crossed with green and wrinkled. Then the genotype, we wrote them at the top, so all homozygous. Then meiosis has to occur so that we can produce the gametes in order for fertilization to occur. Remember to write these two, as you will get a mark for meiosis and fertilization. So how on earth will you get the gametes? Because now there are two traits. So the gamete selection here is going to be a bit more complex than the monohybrid. So using the dominant traits, you need to get, for example, all the possible combinations that can go with this first allele. So this one, the combinations, has to be with the wrinkled or smooth seeds. So in this case, the seeds will be yellow and smooth, the one combination, and then it can also go with that second allele there, but it will also look like that since everything is homozygous. Then you need to do the same for the second allele for color, so that one will be able to combine with that, and same with the second R. So that is what the gametes will look like. What I'll do is I'll write them in different colors, so I'll write the one parent in the one color and I'll write another parent in another color. So let's put all our alleles down here. Okay. Remember I said that there are 16 possible outcomes, so that has to do with this table. Your Punnett square will be much bigger than the previous in the monohybrid, because now you need to show those 16 possible outcomes in your Punnett square. Let's look at the other parents' possible genetic combinations. This G, go with that one or that one. Same for the second. It's going to look exactly the same. Okay, now we need to combine these traits. So when you combine your traits, you can't write it, for example, like that. You need to keep the different types of alleles together. So it's going to look something like that. Okay, keeping them all together. So let's combine these traits now. I'm going to keep it with the colors so that you can see what I'm doing. Remember, this column has to go down, and then I'm writing the capital G first, and then I'm leaving a space for the G that's coming in from the second parent, and then a space for the R from the second parent. So you can do that all the way down. And let's do the other parent now. So going across, these have to be transferred. So that's a lowercase g, lowercase r. Okay, so that is how all of these tables will look because they are homozygous. As soon as they are heterozygous, it's going to look a bit different. So what I'm just going to do is I'm just going to complete these in one color, because I'm sure you paid attention of how I did the cross now. Okay, now we can finally show our F1 generation. So F1 genotype, and it's going to be 16 of these, and then phenotype 16, and then once again you can just use the dominant traits, so yellow and smooth. And then there's no real phenotypic ratio here. So you can also just say 100% yellow and smooth, depending on what they ask of you in the question. So now we've got our F2 generation or our F1 generation, and to get to our F2 we are basically going to cross two of our F1 generations with each other. So it's going to look something like that. Just make some writing space for myself. Okay, let's get started with the F1 generation. So now it's going to be P2, and the parents will be the F1 generation offspring that are now going to get crossed. So the phenotype is they are heterozygous, but we just need to write yellow and smooth crossed with yellow and smooth. And then you can write the genotype. Okay, now we need to get our gametes. So both of the parents will be the same because they have exactly the same characteristics. So I'm just going to do one parent to show the possible combinations that can arise from it. So using this first color, so it is yellow. Yellow can go with a smooth seed or a wrinkled seed. So the allele combination will look like that. Then for the second, which is the green seed can either be smooth or it can be wrinkled. So this is what the green seed combination can look like. And then writing it into the slots of the Punnett square, this is what you will have. And then it will be the same for the other parent. Let's do it in another color. Okay, now you need to combine them. So this block, it will be that parent with that parent, and this block, same thing. So let's do it in blue. So this is what the first combination looks between these two, and then we're going to have to do a combination with these two, and that is how it will look. I'm just going to carry on doing them. We will do this again in class. It's just so much easier having you in front of me so I can see your faces and know where you are snagging with regards to doing the Punnett square. So don't worry if you don't get it as of yet. We will do it again. Remember writing the different allele types together, so you don't get confused and don't write it, for example, like that. Keep the specific groups together. You see how many variations are coming up with the heterozygous one. Okay, so now we've completed the Punnett square. Now we can finally do the F2 generation, and we can start with the genotypes. So what you should do here is just write them all down and then cross them out as soon as, because you also need to count them. So let's find a nice color that will react well on the blue. I think let's stick with red. So the first one And how many of them are there? I only see one of those. So 1 out of 16 will look like that. Then the next one, how many of them look like that? 1, 2. So 2 out of 16. Then the next one, 1. So 1 out of 16. Then the next one, how many of them? 1, 2. 2 out of 16. Next one, how many of them? 1, 2, 3, 4. The next one, how many of them are there? 1, 2. Only see one of those. Do you see why you need to cross them out so that you know what you have done? 1, 2, and then finally that one over there. So 1 out of 16 will look like that. So now you've gotten the genotypes. Now you need to do your phenotypes. And this you do by looking at the dominant alleles only. Okay, so I'm just going to underline all the traits, or, well, not the dominant, but the specific traits that come through. Because if you're just checking at the dominant, you're not going to be looking at, for example, these, and that is wrong. So let's see what we have. So firstly, all the ones that are yellow and smooth, now we need to go count them. So there's 1, cross it out. Then there's 2, 3. Remember, you need to count that. So 3. So we've already got 3. And 4, 5. So plus 2. Plus there's 4 over here. Plus 4. Are there any others? No, that's not one. Okay, so now we just go count it all. So 3 plus 2 is 5, plus 4 is 9. So 9 will be yellow and smooth, regardless of whether they are homozygous or heterozygous. Then the next. So the next one will be yellow and wrinkled that we need to look for. How many of them are there? So there's 1 that is yellow and wrinkled. There's another yellow and wrinkled, and there are 2 of them. And that is all. So in total, there will be 3 that are yellow and wrinkled. Next will be green and smooth. How many of them are there? There's 1, 2, 3. So 3 of them will be green and smooth. And then lastly, green and wrinkled. There will be 1. And this you can then also use to do your phenotypic ratio. So it's going to be 9 to 3 to 3 to 1. Because there will be 9 smooth and yellow, 3 yellow and wrinkled, 3 green and smooth, 1 green and wrinkled. So this is the phenotypic ratio. If you have a dihybrid cross where you are crossing heterozygous characteristics with heterozygous characteristics. So I hope that makes sense. And then you can also go work out the percentages for these if they ask the percentage ratio. I think I've got one more question for you. Yes, I do. Okay. So let's read this one. In guinea pigs, black fur is due to a dominant gene B. So that is for black fur. And white fur due to a recessive gene B. short fur is due to a dominant gene S, and long fur due to a recessive gene, also represented by S. So here you're going to have to write very well, otherwise you can lose marks. A breeder did the following cross. So let's quickly write it. They're going to ask for the genetic cross, and they're going to ask for the phenotype ratio. So the P1 phenotype so you can say it is black fur that is short crossed with white fur that is long. How do I know that? They gave it to me there, and we just did those, what all those letters stand for. So genotype, you can basically just go copy what they have in the question. So meiosis, to produce gametes, and then fertilization can occur. Now, what are the possible gamete combinations? Let's do one for the first parent. So that be, can make sure you write your S's nice and properly, and then the white fur can be combined with short and long fur. So those are the alleles for the first parent. So we can go write it down like that. So you can see that there's a nice difference between my S's specifically. And then for the other parent, which will be all of the alleles will look the same. They're all going to be lowercase b's and s's. Okay, so going down, all of them in this line will look like that. These ones will all look like this. Okay, now we can do the, just want to make sure I wrote F2 there for you guys. Yes. Now we can do the F1 pheno. Let's start with genotype. So 4 of them will be like this. 4 of them will be like that. Okay, then the phenotype. Let me clear that so you don't get confused. So the phenotype, now remember, B is for black fur, lowercase is for white, that is for short fur and long fur. So 4 of them will have black short fur, 4 of them will have black long fur, and 4 of them will have white short fur, and 4 of them will have white long fur. Phenotypic ratio here, there isn't really one because it's 4 to 4 to 4. So it's going to be that. They can ask you the percentage chance. What is the percentage chance of having all of those colors? So percentage chance will be 25%. I'll just write it at the back, 25%. Since all of these together need to make up 100%. So that is the end of the dihybrid crosses.